Okay, great. So good morning, everyone, and welcome to today's session of the Structure Mixed Function webinar series. So before we start, I would like to introduce Instruct for those of you who don't know what it is. So, well, Instruct is a pan-European distributed research infrastructure which provides to the scientific community access to high-end technologies and methods in structural biology. So as you can see here in the map, currently it has 17 members and there are 11 centers providing access to each service. And actually I work in one of them, the CEITEC from Madrid, where is this cryo-EM facility, and I work here as a computer scientist. So well, let's go to the talks then. I'm gonna stop sharing. And well, our first speaker is Jiri Novacek. He's the head of the cryo electron microscopy and tomography core facility of CEITEC, another center of Instruct, placed in Brno. And he's going to give a talk on the platform for scientific data management he and his team have developed, called SIP. He will tell us about its main functionalities and how the implementation was in a mid-size cryo-EM laboratory. So Jirka, now you can start sharing your screen.
All right, thank you. Thanks a lot, Irena. So can you see the slide? Just briefly confirm and then
yes.
Yeah. All right. So thanks for joining. And even though this is a structure meets function seminar series, we'll this time start with a topic that is more technical. And I'll still hope that there will be some structure, but not the structural data in there. So I'd like to present and show to you the platform that we have developed to facilitate the management of the data that are generated in a mid-size EM facility like ours. This topic is in general, I believe, relevant to those of you who are operating the service labs that are not connected to the large infrastructures such as synchrotrons. So when we produce the data for a user in our lab, we typically collect the data that are then shared somehow with the user, who is actually the owner of the data, and they will analyze their data, produce the results, which in a case of the cryo-microscopy will be cryo-EM maps that they will deposit to the database together with the coordinates that they will deposit to PDB through one dep maybe, and that is what they eventually need for publishing. Eventually, they can also publish the raw data following the publication of the results of the data analysis in the database that is focused on a raw data. In the case of the EM, this is Empire. Yet this is not a condition that is generally requested, and since there is additional workload related to that, this does not necessarily always happen. As you can see from this graph, where currently, or at this was done last week. So last week there was almost 40,000 EM maps available in the EMDB, which is the data analysis results repository, whereas that there is less than 2,000 entries in the Empire available. One can argue that for instance, particularly for EM, one data set can generate multiple structures. That is, of course, true, and the case for some, but the ratio here is significantly different and probably does not correspond to solely this fact. Similarly, and then I will now show you the data that was shown to me 3 years ago by Jose Maria Carrazzo. Similarly, during the SARS-CoV pandemics, the EM community has produced large number of structures, but only for very few of them the original data were published together with the structural data itself. So what we are trying to suggest and implement is the slight adjustment to that original data workflow, and that contains additional parts that allow us to preserve and transfer or hand over the data to everyone who needs the data in a more or in an easier way. So we are working with a cloud platform to which we actually upload the data directly from our instruments. Those data are made available to the user, to the owner, such that they can analyze them and then again publish the results. And we have developed tools that allow us publish the data or the metadata and the links to the original data upon either user request or upon the expiration of the embargo period. Primarily, we are not, in our case, working with a database that is dedicated for EM data, which is Empire, but that is something that I'll show in the end as an outlook, and we are using a more general database for the scientific data that is available. So, in brief, what do we need to do when we operate a facility and collect the cryo-EM data, or how does the specific cycle of the cryo-EM data look? As with any other imaging data, the cryo-EM data are typically very heavy on the size of the original raw data, because we're acquiring images, and per data set we usually have something like 1 to 2 terabytes of the raw images. So this is produced by an electron microscope. Then ideally we would have a computational infrastructure near the microscope that would allow us to do the real-time analysis of the data, such that we can get a feedback directly whether the quality of that data is sufficient or indicates indication that the data quality is good, and subsequently transfer the data to the storage through which we can make the data discoverable by the owner. Eventually, this poses yet another need for the facilities to have not only the instrumentation, but also a particular infrastructure for the data processing and data storage. So with that in mind, we set a couple goals when trying to develop an alternative system that would leverage the benefits of the fact that there are potentially other experts in the computation and data storage. But most importantly, we thought that the system that we want to implement needs to be easy to set up and easy to operate. It should not be platform independent, and it shall be also hardware independent. Of course, the hardware would influence the performance. We eventually thought of the workflow in a general way, such that it can be used not only to the cryo-EM data, that is our field of interest, but to any kind of data coming from different research infrastructures. But the cryo-EM data pose a typical or one domain that is very heavy on the size or size of the initial data, and thanks to that, it is one of those cases which might require extra attention, and managing those should show that handling other types of data that are not that heavy or sizey might actually be easier and manageable with the same system as well. So what we want to do for the EM data in particular is that we want to do the data annotation directly on the instrument, and then deployment of the data directly from the microscope to the certain cloud, and automate the archival and publication processes. Since we want to get rid of the necessity of running the real-time data analysis on our resources, we decided to join with the HPC center and carry out the data there. So here is the graphical display of the system that we've developed and we are now using. So we have our microscopes that are in the internal network, not accessible from the internet, and within that network we have a single instrument that has a secured connection to the particle web server. The web server is an orchestrator of the whole system and communicates with individual components, providing tasks to be carried out by that particular component. So the first step is a data transfer from the microscope. The data is transferred to the cloud provider. So we use iRODS, federated cloud solution, as a platform to transfer the data around in our system. And once the data are transferred to this cloud storage that is close to the HPC, we can start the data analysis. For that, we've implemented two solutions. One is based on Scipion, and the other on CryoSpark. Both things were very popular in the community to run the real-time data analysis. Once the data analysis is finished, or directly at the start of the experiments, the links or the way how to get to the data are shared with the user via email, and the user can either start working on the same HPC if they have access there, or download the data from there and continue the data analysis on their own. Eventually, we use the EUDAT services such as BPIT and B2Share to assign a persistent identifier to each data set. So with this, each data set is assigned with a PID or DOI, and then it can be made publicly available, or particularly the metadata that we collect are made publicly available through the B2Share. So now for some details how this works, and I will go quickly through it. As mentioned, everything is orchestrated via a single web server. to which the user identifies through a third-party identity provider. So we use either OrcID for the academic users or Google for the non-academic users. And that web server provides an interface to the graphical user interface that you can use to interact with the whole system. That web server also in parallel is an orchestrator and communicates with all of the nodes. Such a node might be either storage infrastructure, such a cloud storage, or the computational infrastructure, such as the computational resources, or the front end computational resources on the HPC, or actually the service lab. So the EM lab from which we collect the micrographs that are then submitted to the system. The web server can actually single web server can actually operate multiple labs. So that allows us to scale up and facilitate the utilization of the computational resources, because we can have multiple labs like ours connected to the single web server and utilizing the same storage and computational resources. A bit of a detail how this works and what are the requirements. So the only requirement from the network perspective is that each of the nodes that we call lymph nodes needs to have HTTP access to the web server, to the orchestrator, and the communication is only one way from the node to the server. The individual nodes are then handled by polling. As for the data itself, so the different nodes, the different lymph nodes have different roles. In this particular case, we have one node that actually pulls the data from the microscopes and pushes it, transfer it to the storage. We've implemented different type of storages. So one can use the standard physical storage or storage that you can mount to your system and transfer the data via defining a particular path. And we've also worked with two different cloud solutions. The primary one is iRODS that we primarily use, and the secondary that we've tested earlier but abandoned later on was one data. And here is how this actually looks. So we are operating in Brno, which is one city in Czech Republic, where we have the cryo-EM lab. And the data is actually transferred to the HPC center, which is in Ostrava at the IT4I. I mean, physically this is 170 kilometers, but in practice, what's more important is that there is a good network connection between the two nodes, and including the last miles, as this is called, the network connection from the microscope to the HPC is always 10 gigabit or better. The spine network in our case is 40 gigabit. So the utilization of these resources removes the necessity of us having the computational infrastructure here. And as mentioned before, we could easily connect multiple centers like ours to this single infrastructure as well. minimizing the needs of having the additional computational resources scattered across the labs. So this, I mean, even though this is not physically close, it allows us to perform on-the-fly data analysis, and we have two different workflows available. One for CryoSpark, where we use the standard CryoSpark live workflow, and the other for Scipion, that for which we offer a predefined workflow, but we also have an option to download new workflow that is validated from the workflow hub. So a user can eventually design her or his workflow that is then uploaded to the workflow hub and can be directly used during the data processing. Currently, we use a standard queuing system on that HPC without priority for particular priority for the facility, and for the EM data, we can show that we actually can easily transfer them between these two centers within the time that is in general below 2 seconds, as shown for these almost 20,000 movie images, bearing in mind that each of them has some 150 to 300 megabytes. This time allows us to actually perform the data analysis in real time. And with that also obtain or receive back the feedback referring on the data quality that allows us to either interfere with the instrument or not. So I think that I should speed up. So we actually differentiate data into 3 types. The ones that are main for archival, the second that are alive just for the one data analysis campaign, which is the acquisition and analysis of the data set, and the third, which are temporary and are removed directly once they're after they are created and analyzed. So eventually, we aim to archive the initial raw data and the metadata, the workflows that are used for the data processing, and eventually the results of the, of course, of the data processing. How individual nodes work. So each of these nodes is implemented in Python, so it can run on both Windows and Linux systems, and it is meant to operate continuously. So being a part of, for instance, a service task that is run on that particular instrument. They communicate with the orchestrator, and based on that communication, they spread individual tasks to either their resources or carry out the task on their own. So now more from the user perspective, what I will try to show you is that even though this is now set up for the EM facility, it can be easily changed to any other type of a facility. So the standard configuration is performed through a single configuration file. We use a YAML format. So it is a single YAML format that configures how the facility and all its resources are set up. So for each facility, we can define multiple instruments, and if needed, each instrument can have its own workflows. These workflows are particular workflows related to data analysis. So in the case of EM, this would be either single particle or tomography or electron diffraction, for instance. One can easily imagine that this could be just the data transfer. That is the simplest workflows, and that can be implemented for any type of the solution and facility. And this is how the front-end application looks. So it is web browser based, so which means that the user do not need to install any additional software. It is accessible directly from the microscope, and it is actually the single point how to transfer the data from the microscope to the user. With that, we enforce that at least some minimal metadata are filled in before the data can be transferred from the microscope, and thus we generate a minimal data model that goes together with the data. The data model that we are working with currently has 4 components. It is the information about the operator and the center, so about our lab and about the person who actually is responsible for acquiring those data. Then it has an information about the user, so the data owner. We also collect some very general information about the sample, and then the information about the data itself. Our data model for the data part is actually inspired or corresponds to the ISPy-B data model that is used at some synchrotrons for cryo-EM data acquisition. Once we start the job, the data are submitted to the cloud and analyzed, and we are getting a report on the screen with the updated data status, which allows the user to check if the quality is sufficient or there is an intervention needed with the instrument. In parallel, the user is notified by an email, and within that email, she or he gets a link to the project web page, and on that web page, we share the information about the data acquisition, so the setup of the data acquisition, the report which was available on the previous slide, and also the information how to access the data, whether that data has been selected for long-term preservation, so for archival, and if that is the case, also a button to submit or publish the data through the public database, the B2 or UDAN databases in our case. Apart from these features, the data management lifecycle, we during the time added additional apps to our system that from which I would primarily mention one that I found very useful. But we also have an app for the sample storage, so sample management, the booking table, the visual inspection of the time plan for the particular instrument and particular date. And this is very specific to EM. We also keep the information about the autoloaders content for the autoloader systems, and the current status, history, and time for which the individual grid samples been on a stage. But the most important part of this additional apps is the remote access. So through this application, we allow the users to access the instruments remotely from their home. So I already mentioned that they identify against the system using their own identity, org ID primarily. And whenever they have a session planned in the planning board, they can access the particular instrument that they have a session or on. The remote access, for the remote access, we use the Apache Guacamole. That means that we need to have either VNC or RDP remote desktop available on that particular instrument. And that's it. Otherwise, the Guacamole runs in the browser, which means that you can access it directly either from any PC or from your mobile phone without the need of additional software installed. This allows the user to either set up the session remotely or check during the acquisition the status of the instrument. And of course, for the administrators or the facility people, it allows to fast access to the instrument at any time to intervene if there are particular problems. So I will conclude with that. So we have put together a workflow that facilitates the life cycle of the cryo-EM data. It's composed of the part that automates the annotation of the data. It's transferred to the cloud storage, on-the-fly data processing, and eventually publication of the raw data in a state together with the metadata in a state that is closer to what one could call a fair principles. Eventually, we would also like to use this information, the metadata that we have, and the data from the data processing to run a feedback loop. That is something that we are working on, and eventually also initiation of the deposition to the public databases. And with that, I would like to thank particularly Radek, who designed and is working on the whole application. And this is a collaboration with Martin Golasowski and Jan Martinovic from the IT4I. There's also a collaboration from Irene, Carlos Oscar, Carolina, and Jose Maria from the CNB in Madrid. And with that, I want to thank you for your attention.
Thank you very much, Erika. A super interesting talk, especially thinking that this can be used in other techniques apart from cryo-EM. So congratulations. So maybe we can have one or two questions. I don't know. I don't see any questions on the panel. If there is no questions, I will go for one. So, well, so the first one is, Erika, this is currently being used at CEITEC, right? So,
yeah.
Is okay. And one thing is related to the connectivity between the web server and the other nodes. It's correct that the other nodes doesn't need to have any open port, just to go to perform HTTP request to the web server, and with that will be all, or
yes. Yes. So yeah, it is only that port that would allow for the HTTP sending the HTTP requests. For the microscopes, in our case, we have all microscopes in one local network, and using one PC in there that we call support PC to actually perform all the requests from all the microscopes. So then these send the request through the support PC. Eventually, for such a closed networks, you can get along by having one restricted connection to the internet, and with that communicate with the orchestrator.
Okay, thank you very much, Jiri.